This is the Gut Microbiome Project. I will email you a specific, the project just so you can read it all. This is our abstract. I will start by defining what microbiome is. So microbiome is the types and proportion of microorganisms living in a given location. That's like the broad definition. A gut microbiome, which is a little bit more specific, is the study of these microorganisms, specifically in the intestines of certain organisms. And this is what we are actually testing in this project, is the gut microbiomes of 4 different species, or as we use dogs, and what is correlated to their health and what kind of bacteria there is related to it. So I will start with saying that the gut microbiome is correlated to diet, the environment, and several aspects of health, including disease state, medications, body weight, and host genetics aspects of health. So over here, we actually looked up 2 types of research that were correlated to the gut microbiome of an animal. So basically, research suggests that a human gut microbiota has a significant impact on cardiovascular diseases, including heart failure and diabetes. So that's just a research that we believe that it was like actually close to our project. Um, so So the bacteria present in microbiomes is highly associated with the pathogenesis of insulin dysfunction and T1D, and targeting gut microbiota may serve as therapeutic potential for T1D patients. Han. So studies have showed that at the phylum level, there's an abundance of actinobacteria and firmicutes, and the ratio of firmicutes to bacteroidetes were all lower in the children with T1D than the healthy children. Studies have shown that at the genus level, healthy children have an abundance of Lactobacillus, Bifidobacterium, Blaudia coccidis, Eubacterium rectale group, and Prevotella in the gut, while children with T1D contain greater numbers of Clostridium bacteroides and Veillonella. This research shows a causal link between the gut microbiota and the onset of type 1 diabetes. So this is basically talking about the gut microbiome related to type 1 diabetes in children. So basically it's saying that it's related to the pathogens of insulin dysfunction in T1D. Okay. And the second research is actually highlighting the importance of antibiotics on gut microbiota. So basically it is saying that it is highly associated with body weight, as the use of antibiotics can cause a decline in bacterial diversity. So it has been linked to weight gain. Other studies have shed light on gut microbiota and its increasingly more substantial role in metabolic disorder and the development of food absorption and low-grade inflammation. This research also suggests a causal link between the gut microbiota affecting this health factor of body weight and, more specifically, weight gain or obesity. So all of these factors affecting the gut microbiota and vice versa. Does gender affect the gut microbiome of similar organisms? This was actually our question for this project, and our hypothesis was that a change in gender of relatively similar organisms, such as species, weight, breed, and age, will have similar microbiomes. So this was a very broad hypothesis, but we actually just decided to have 4 different organisms that had different genders, like 2 different genders, like 2 females and 2 males, and they would be closer in health and like maybe environment, and see what could actually cause a difference in their gut microbiomes, so we can see if there is a change in their weight, breed, or age will have similar microbiomes or not. Okay. So the relationship between the gut microbiomes and physiological conditions, such as over here the body weight, or like antibiotics affecting the body weight, and then all Alright, so for our materials and methods. So during our experiment, we used samples of 4 different German Shepherd species. We used 2 years and 6 months old male that weighed 80 pounds, a 1 year and 4 months old male that weighed 67 pounds, a 1 year and 2 month old female that weighed 70 pounds, and a 4-year-old female that weighed 85 pounds. So the majority of the samples we used were close in age. They had like weight and fecal matter that was kind of similar. So we then weighed the fecal matter and added 1 milliliter of inhibit X to each tube and homogenized each sample and then incubated it for 5 minutes and 70 degrees Celsius. We centrifuged all the tubes at 14,000 RPM for 1 minute. And then pipetted the supernatant and added it to new tubes with 15 microliters of proteinase K. So proteinase K basically breaks down any kind of contaminants such as proteins and stuff like that. So then we added 500 microliters of buffer AL to each tube, and then we vortexed it for 15 seconds. After that, we incubated the tubes for 10 minutes at 70 degrees Celsius, and then we transferred 500 microliters of ethanol to the lysate, and we vortexed. So ethanol is used to basically wash any contaminants or stuff like that while we're going. We then transferred 750 microliters of the lysate into the Kyogen spin column, and we centrifuged each column for 1 minute at 14,000 RPMs. And then we discarded the filtrate. Afterwards, we added 500 microliters of wash buffer AW1, and then we centrifuged for 1 minute at 14,000 RPM. And then we discarded the filtrate, and then we repeated this process and centrifuged for 3 minutes at 14,000 RPMs. Then we transferred the columns into new centrifuge tubes, and we added buffer ATE into the Kyogen spin column. We incubated at room temperature for 1 minute, and then we centrifuged for 1 minute, and then kept centrifuge tube while eluted the DNA on ice. It's always important to leave your DNA on ice because it will help it stay uncontaminated. So we then added 150 microliters sample and 400 microliters chipped DNA binding buffer. We vortexed and centrifuged briefly, and then we transferred the centrifuge for 30 seconds. The centrifuge samples into label zymospin columns. So just so I can clearly explain, so Kyogen spin column is used to separate the DNA from the scat sample that you get at first. It is a commercially available kit. It separates your scat sample from the DNA, so it will have the DNA out. And then we use a zymospin column, which is another commercially available kit, to concentrate our DNA tenfolds. So we use a zymospin column and place 2 milliliters into the collection tube, and we centrifuge for 30 seconds at 13,000 RPMs. So lastly, we added 200 microliters DNA to the buffer to washing column, and we centrifuged for 1 minute, and we repeated it twice. So after purification, concentrate the DNA, in which we use a nanodrop according to the directions given by the manufacturer. The nanodrop also measures the absorbance, and it also calculates the concentration of the nucleic acids and purified proteins. We amplify the variable region of the 16S ribosomal RNA gene by PCR. Amplify the variable region of the 16S ribosomal RNA gene by PCR. So this was our main goal, is to amplify the 16S RNA purified, like to purify that ribosomal RNA gene to because it's related to microbiome of an animal. So we then use gel electrophoresis, which usually the molecules migrate through the gel like matrix. so they can be localized following electrophoresis. The gel electrophoresis uses 250 volts. Then we use the AgentCore Ampere XP kit. So before I keep going, gel electrophoresis, what happens is DNA moves from the cathode to anode because, as you know, DNA is negatively charged. So we use gel electrophoresis to see if our DNA is actually working. So we put all 3 samples, and unfortunately one of our samples did not work. So we actually just kept going. So we had 3 samples total. But it was okay since we had enough, like we have 3 successful DNA samples. So we use the AgentCore Ampere XP kit for our PCR cleanup. This kit is used to clean up any contaminants after the PCR reaction. It helps remove literally all the amplicon. So the kit is used to clean up the amplicon from the PCR reaction. Then we use the fluorometer to find the index PCR concentration. So what we did is, spectrophotometers and fluorometers have one difference. Actually, they have a couple differences, but the main difference is that a fluorometer actually can find the, can differentiate between DNA and RNA, which is cool because the spectrophotometer does not. Also, a fluorometer is more accurate, I believe is less time consuming, and it's very useful with accuracy. You know, it gives you accurate results. So we use the fluorometer in that case, not a spectrophotometer, due to it being able to differentiate between RNA and DNA. And then we had to do some conversion problems to find the molar concentration. So we actually had to figure out the concentration up here and the 2060-2080 ratio as well. So moving on to our results. So for DNA sequencing, which was a part that we actually did not do. We just finished our samples and we had to send it down to be sequenced. DNA sequencing is actually used by the MiSeq tool. So that's what sequences it. And then we also had, okay, sorry about that. So our results up here, they start over here. I will move as I speak through it. It's not in order. So I'm going to speak about the nano drop first, since that's the first thing that we did in this process. So the nano drop basically tells you, as I said again, it tells you the concentration of nucleic acids and purified proteins. So these numbers will tell us how much is, like, you know, nucleic acids and purified proteins. So we had the 4 samples. So sample number 406 had 23.8 nanograms per microliters as a concentration and a 260-280 ratio of 1.77, which was great. Sample number 521 had a concentration of 13.6 nanograms per microliters and a 2060-2080 ratio of 1.58, which was relatively lower than we wanted, but it was still okay to work with. 643 had a concentration of 13.1 nanograms per microliter and a 2060-2080 ratio of 1.89, which was great. And then sample number 1048 had a concentration of 16.4 nanograms per microliter and a 2060-2080 ratio of 1.92. So concentrations were all good. According to our lab protocol, we at least had to have 6.7 nanograms per microliters to be suitable. Our ratios were all very close to 1.8, 1.8 being pure DNA. We actually had one hit 1.89, which was actually very good. 1.77 was really close to 1.8. 1.58 was the lowest we had. As I said, we wanted it to be better, but we can still work with that because DNA purity is not just 1.8. I mean, this is completely pure DNA, but between, like, I think it was like 1.5 and 1.8, you should be fine. It would have like a slight contamination of like proteins, but it shouldn't stop you from being able to keep going. So after the nano drop, okay, nanodrop down to gel electrophoresis. So gel electrophoresis, this is another group's result. You will see it in the PowerPoint more specifically, but I will just explain it. So down here is our results. Up here is another group's results. Our positive control was very bright, which is good. Our negative control was not there, which was what we expected. For our sample 521, it was pretty low. Sample 406 was pretty good. 1048, we did not find anything. And then 643 had a light, like a lower light. So that was pretty great. Our results were what we expected. Right? So we used a nanodrop in the first place because we wanted more accurate results. It gives you the most accurate results. Alright, so the positive and negative controls show that sample 406 worked very well because at the same length as the positive. Control was also bright, and 521 and 643 had a good length but not as bright, so not as much DNA was present. And then we excluded 1048 because it did not show anything on the gel electrophoresis. So the results of the fluorometer over here. So 1.6 micrograms per milliliter is a good concentration. All of ours were very low. We knew this at the point that we couldn't do dilutions with our DNA because they were just not good enough. So we just kind of stopped over here and just stayed with our results. But we did get them sequenced anyway because they weren't that bad. They were just not enough to make the dilutions, but we still could sequence them. So then our samples were not sequenced due to a machine breaking, so we did not get any results. And so we had to use another class's analysis from last year, which was okay. That's what we actually did. So we replaced 1048 with 365, and then it was a male replacing a female, which is not good for our hypothesis, but we can still compare the male microbiomes to each other, and then we can compare over here, the samples were pretty close, so it's fine. We can still compare the male microbiomes to each other and then compare them to single female, which should be fine for us. So kingdom, order, and genus classifications down here, they all show how their microbiomes are similar. So kingdom level being the highest, and then genus would be lower. Species would be lower, then genus would be the lowest. So kingdom, order, and genus were all showing that the female sample 406 had a type of bacteria in common with at least one of the males. So the female actually had something similar with each male, which was really good because it supports what we wanted. So so the bacteria that was in all species were Prevotella copri, and that could possibly be a trend for the gut microbiomes of all dogs of this species. So over here, the Fusobacterium and the Megamon bacteria for 406 were actually very high compared to sample 365. We were actually worried at first for that, but then we looked it up and it was related to soil. So it shouldn't be bad because it means that maybe the female's environment had a lot of soil. So that's why her numbers were very high. We thought it was something related to her genes, which was not. So that was a good thing because that means it's just correlated to health. In the species, they were actually all pretty close except for sample 521, which is a male. It was 17.39. It was a Prevotella copri, but they were all very close, so it wasn't really significant for us to think that it could relate to anything. For the kingdom, they were actually all similar, like identical, except for sample 521 being 99.05, which is totally fine. It's like 0.8%, which should not like be an issue. And our positive control matched all of them. Anything that's called unclassified or other in these charts means that the bacteria was either not relevant to our project or that it was just not yet discovered. So if you see that, it does not mean that it's something else. Alright. Okay, so moving on to discussion. I will just move this over. So discussion is over here. I'm going to move over. So we extracted DNA from the scat by using 2 commercially available kits. We use a Kaigen kit to isolate DNA from scat, and we use the Zymo kit to concentrate this DNA tenfold. So DNA extraction was successful because we had a good quantity. Our UV spectrophotometry result for sample 1 in the 260-80 ratio was 1.58. This indicates that it has little too much protein because DNA purity between 1.5 and 1.9 is like, pure. Sample 2 had a ratio of 1.77, which indicated that it had a good DNA purity. Samples 3 and 4 both had too much RNA in them of concentrations 1.92 and 1.89. So during our amplicon PCR in the gel electrophoresis, we expected a bright light or a bright line for our positive control, which is what we got. For the negative control, we expected no line to occur, and that was what we got. We expected a line for all 4 samples, but we unfortunately did not get one for our 3rd sample. So this basically explains that the PCR amplification was not successful for our 3rd sample. So we continued with other 3 samples and our positive and negative controls only. The presence of a PCR band means that our PCR amplification was successful. So when you see a band in your gel electrophoresis, it means that our PCR amplification, worked. So if you see it in gel electrophoresis, it means that it's correct or it's successful. So our results for the index PCR cleaning for the negative control was an absorbency of 0.028 nanomolar, and our positive control was 0.623 nanomolars. So from this point, we're actually interpreting results from different animal fecal samples because the results from our original samples were unavailable due to a broken machine, so we didn't get our results. So we had 11,062 reads in our analysis over here in the positive control. This result tells us that our positive control had a very high DNA purity and that cross-contamination was like very low or almost not happening. Our first sample, 365, we had 14,301 successful reads. Sample 406 had 13,164 successful reads. Sample 521 had 18,984 successful reads, which was our highest. Sample 643 had 10,633 successful reads. And yeah, so samples with reads above 1,000 means that they are reliable. So we had lots of DNA. So for 521, it's like super reliable for us. So they were actually all higher than 1,000, which was pretty much very reliable. So even though the gut microbiomes of our organisms are at such a specific classification level, the most abundant bacteria in each sample was actually Prevotella cocci in male and female. So this shows that the most bacteria present in the fecal samples were identical at the species level, regardless of gender. Sample 365 was a male, and sample 406 was a female, and they were almost identical with the types of bacteria expressed at each classification level, with the exception of the Fusobacteria and Megamonas down here being a little weird between 365 and 406, but that was related to soil, which was related or correlated to the environment of the female. Other than that, they were pretty much identical. Prevotella species are identified to be physiologically able to prevent complications associated with a genus of bacteria in all samples. It can also be used to explain the similar physiological effects that the species of organisms may have regardless of gender. And our hypothesis was that a change in gender of relatively similar organisms, like species, height, and breed, will have similar gut microbiomes. Our question was if gender affected gut microbiomes of similar organisms. Our hypothesis was supported because there was no change in the gut microbiomes of similar species of opposing genders, with the exception of the change with the fusobacteria and the megamom. We did not believe it was very much a difference. It was just a difference between environment. It wouldn't affect our hypothesis, in our opinion, but yeah. So this was the project. I will email you the project just so you can read through it and the abstract. Thank you.